Welcome to Chapter 2 of Class 12 Biology, Human Reproduction. In this comprehensive chapter, we'll explore the fascinating biological processes that enable human reproduction, from the formation of gametes to the birth of a new human being. Humans are sexually reproducing and viviparous organisms, meaning fertilization occurs internally and the embryo develops within the mother's body. The complete reproductive sequence includes gametogenesis, the formation of sperm and eggs, insemination, the transfer of sperm into the female reproductive tract, fertilization, the fusion of male and female gametes to form a zygote, implantation, the attachment of the developing embryo to the uterine wall, gestation, the period of embryonic development, and parturition, the process of childbirth. There are remarkable differences between male and female reproductive processes. While sperm production continues throughout a man's reproductive life, egg formation ceases in women around age 50. Understanding these processes helps us appreciate the incredible complexity and coordination of human reproductive biology. Let's begin with the male reproductive system, which is located in the pelvic region and consists of several key components: a pair of testes, accessory ducts, glands, and external genitalia. The testes are the primary male sex organs responsible for sperm production and hormone secretion. They're situated outside the abdominal cavity within a pouch called the scrotum, which plays a crucial role in maintaining the temperature to -2.5 degrees Celsius lower than normal body temperature, essential for proper spermatogenesis. Each testis is oval-shaped, about 4 to 5 centimeters long and 2 to 3 centimeters wide, covered by a dense protective covering. Internally, each testis contains approximately 250 compartments called testicular lobules. Within these lobules are highly coiled seminiferous tubules where sperm production actually occurs. The seminiferous tubules contain two main cell types. Male germ cells are spermatogonia that undergo meiotic divisions to form sperm, and Sertoli cells that provide essential nutrition to the developing germ cells. Outside the tubules in the interstitial spaces, we find Leydig cells that synthesize and secrete testosterone and other androgens. Now let's examine the male accessory ducts and glands that complete the male reproductive system. The male sex accessory ducts include the rete testis, vasa efferentia, epididymis, and vas deferens, forming a continuous pathway for sperm transport. The seminiferous tubules open into the rete testis, which connects to the vasa efferentia. These small ducts leave the testis and open into the epididymis, located along the posterior surface of each testis. The epididymis serves as a storage and maturation site for sperm before they move into the vas deferens, which ascends into the abdomen, loops over the urinary bladder, and joins with the duct from the seminal vesicle to form the ejaculatory duct that opens into the urethra. The urethra then carries sperm through the penis to the external opening called the urethral meatus. The male accessory glands include paired seminal vesicles, a single prostate gland, and paired bulbourethral glands. Their secretions constitute the seminal plasma, rich in fructose for energy, calcium, and various enzymes. The bulbourethral glands also provide lubrication. The penis, as the external genitalia, contains specialized tissue that enables erection for successful insemination. Let's now explore the female reproductive system, which is structurally and functionally integrated to support the entire reproductive process from ovulation to childcare. The system consists of a pair of ovaries, oviducts, uterus, cervix, vagina, external genitalia, and mammary glands. The ovaries are the primary female sex organs, producing both the female gametes and steroid hormones. Located on each side of the lower abdomen, Each ovary is about 2 to 4 centimeters long and connected to the pelvic wall and uterus by ligaments. The ovarian structure includes a thin epithelial covering enclosing the ovarian stroma, which is divided into a peripheral cortex containing the developing follicles and an inner medulla. The female accessory ducts include the oviducts or fallopian tubes, uterus, and vagina. Each fallopian tube is 10 to 12 centimeters long, extending from the ovary to the uterus. The part closer to the ovary is the funnel-shaped infundibulum with finger-like fimbriae that help collect the ovulated egg. The infundibulum leads to the wider ampulla where fertilization typically occurs, followed by the narrow lumened isthmus that connects to the uterus. Continuing with the female reproductive structures, let's examine the uterus and external genitalia. The uterus, also called the womb, is a single, inverted pear-shaped organ supported by ligaments and opening into the vagina through the cervix. The cervical canal together with the vagina forms the birth canal. The uterine wall has 3 distinct layers: the external thin membranous perimetrium, the middle thick layer of smooth muscle called myometrium that exhibits strong contractions during childbirth, and the inner glandular endometrium that lines the uterine cavity and undergoes cyclical changes during the menstrual cycle. The female external genitalia include the mons pubis, a cushion of fatty tissue covered by skin and pubic hair, the labia majora, fleshy folds extending down from the mons pubis, the labia minora, paired folds under the labia majora, the hymen, a membrane that may partially cover the vaginal opening, and the clitoris, a sensitive finger-like structure. It's important to understand that the presence or absence of the hymen is not a reliable indicator of virginity, as it can be broken by various non-sexual activities. Now let's dive into gametogenesis, the process of gamete formation. In males, this is called spermatogenesis and occurs in the seminiferous tubules of the testes, beginning at puberty. The process starts with diploid spermatogonia containing 46 chromosomes that multiply by mitosis. Some of these develop into primary spermatocytes that undergo the first meiotic division to form 2 equal haploid secondary spermatocytes, each with 23 chromosomes. These then complete the second meiotic division to produce 4 equal haploid spermatids, which are transformed into mature spermatozoa through spermiogenesis. The mature sperm become embedded in Sertoli cells before being released through spermiation. This entire process is initiated at puberty due to increased secretion of gonadotropin-releasing hormone from the hypothalamus, which stimulates the anterior pituitary to release FSH and LH. LH acts on Leydig cells to stimulate androgen production, while FSH acts on Sertoli cells to support sperm maturation. Let's explore oogenesis, the female gamete formation process that differs significantly from spermatogenesis. Oogenesis begins during embryonic development when millions of oogonia are formed in the fetal ovaries. No new oogonia are added after birth. These cells start meiotic division but get arrested at prophase I, becoming primary oocytes. Each primary oocyte becomes surrounded by a layer of granulosa cells, forming a primary follicle. A remarkable number of these follicles degenerate between birth and puberty, leaving only about 60,000–80,000 primary follicles in each ovary at sexual maturity. The primary follicles develop into secondary follicles with more granulosa layers and a new theca, then transform into tertiary follicles characterized by a fluid-filled cavity called the antrum. It's at this stage that the primary oocyte completes its first meiotic division through unequal division, forming a large haploid secondary oocyte that retains most of the nutrient-rich cytoplasm and a tiny first polar body. The tertiary follicle becomes a mature Graafian follicle that ruptures during ovulation to release the secondary oocyte. Now let's understand the menstrual cycle, the reproductive cycle in human females that repeats approximately every 28 days. The first menstruation at puberty is called menarche, while the cessation of cycles around age 50 is menopause. The cycle consists of 4 main phases. The menstrual phase lasts 3 to 5 days and involves the breakdown of the endometrial lining, resulting in menstrual flow. This occurs only if fertilization doesn't happen. The follicular phase follows, during which primary follicles grow into mature Graafian follicles while the endometrium regenerates. The ovulatory phase around day 14 features a surge in luteinizing hormone that triggers ovulation, the release of the secondary oocyte. The luteal phase sees the remaining follicle transform into the corpus luteum, which secretes progesterone to maintain the endometrium for potential implantation. If pregnancy occurs, the corpus luteum continues functioning. If not, it degenerates, leading to menstruation and a new cycle. This entire process is regulated by complex interactions between pituitary and ovarian hormones. Let's examine fertilization and implantation, the critical processes that begin pregnancy. During sexual intercourse, semen is deposited in the vagina, and motile sperm swim through the cervix and uterus to reach the ampullary region of the fallopian tube. The ovulated egg is also transported to this region, and fertilization can only occur if both gametes arrive simultaneously. When a sperm contacts the zona pellucida of the egg, it triggers changes that block additional sperm entry, ensuring only one sperm fertilizes the egg. The sperm's acrosome secretions help it penetrate the egg, inducing completion of the second meiotic division and formation of the second polar body and haploid ovum. The fusion of sperm and egg nuclei creates a diploid zygote with 46 chromosomes. This is also when the baby's sex is determined. Since all eggs carry an X chromosome, the sperm determines whether the zygote will be XX female or XY male. The zygote then begins meiotic division through cleavage as it moves toward the uterus. Following fertilization, the developing embryo undergoes remarkable transformations. The zygote divides to form 2, 4, 8, and then 16 cells called blastomeres. The 8 to 16 cell stage is called a morolla, which continues dividing and transforms into a blastocyst as it reaches the uterus. The blastocyst has an outer layer called the trophoblast and an inner cell mass. The trophoblast attaches to the endometrium while the inner cell mass develops into the embryo proper. This attachment and embedding process is called implantation and marks the beginning of pregnancy. After implantation, finger-like chorionic villi develop on the trophoblast and interdigitate with uterine tissue to form the placenta, the vital connection between mother and developing embryo. The placenta facilitates oxygen and nutrient supply to the embryo while removing waste products. It also functions as an endocrine organ, producing hormones like HCG, HPL, estrogens, and progestogens that are essential for maintaining pregnancy. Let's track the embryonic development month by month throughout the 9-month human pregnancy. At 1 month, the embryo's heart forms and becomes detectable. By the end of the 2nd month, limbs and digits develop. At 12 weeks, marking the end of the 1st trimester, most major organ systems are formed, with limbs and external genital organs well developed. The 5th month brings the first fetal movements and appearance of head hair. By 24 weeks, the body is covered with fine hair, eyelids separate, and eyelashes form. At 9 months, the fetus is fully developed and ready for delivery. Meanwhile, The inner cell mass differentiates into 3 germ layers: the ectoderm, endoderm, and mesoderm, which give rise to all adult tissues and organs. This period also involves stem cells from the inner cell mass that have the remarkable ability to develop into any tissue type. The entire developmental process represents one of biology's most incredible transformations. Finally, let's explore parturition and lactation, the completion of the reproductive journey. Parturition, or childbirth, occurs after approximately 9 months of gestation and is induced by a complex neuroendocrine mechanism. Signals originate from the fully developed fetus and placenta, triggering mild uterine contractions called the fetal ejection reflex. This stimulates oxytocin release from the maternal pituitary, which causes stronger uterine contractions that further stimulate oxytocin secretion, creating a positive feedback loop that culminates in the baby's expulsion through the birth canal. Soon after delivery, the placenta is also expelled. Meanwhile, the mammary glands differentiate during pregnancy and begin milk production through lactation. The initial milk, called colostrum, is particularly important as it contains essential antibodies that provide the newborn with crucial immune protection. Breastfeeding during the initial growth period is medically recommended for optimal infant health and development.